Hello, my name is Joshua Mugabe. I welcome you to Blood Types. What are the main human blood group systems? The human main blood group systems are A, B, O systems, and the Rhesus system and the MN system. Why is the determination of the blood types of the donor and of the recipient important in the transfusion? Red blood cells have different antigens and on the outer surface of their plasma membrane. For example, antigens A and B of blood group system ABO are glycoproteins of the membrane. If a donor has red blood cell with antigens not present in the red blood cell of the recipient, that means lacking a transfusion compatibility, the immune system of the recipient recognizes these molecules as actual antigen. That is, foreign substances, and it triggers the defense response, producing specific antibodies against these antigens. The transfusion of red blood cells then are destroyed by these antibodies, and the recipient individual may even die. What are the antigens and the respective antibodies of the ABO blood group system? The ABO blood system includes the erythrocytic antigen A and B that can be attacked by antibodies anti-A and anti-B. The antigen A and B are glutinogens, and the antibodies anti-A and anti-B are agglutinins. What are the blood types of ABO blood system? The blood types of ABO blood system are type A, type B, type AB, and type O. What are the antigen and antibodies of each blood type of the ABO blood system. Type A has antigen A and antibody B, anti-B. Type B has antigen B, antibody anti-A. Type AB has antigen A and antigen B, does not produce antibody A, neither antibody B. Type O does not have antigen A, neither antigen B, but has antibodies anti-A and anti-B. Obviously, antibodies are made by B lymphocytes, not by red blood cells. What is the logic of the transfusion compatibility concerning the ABO blood group system? Transfusion compatibility of ABO system takes into account the antigens present in the red blood cell of the donor and the antibodies that the recipient can produce. Whenever the recipient is not able to produce antibodies against antigen of the red blood cells of the donor, the transfusion is compatible. So, regarding ABO compatibility, type A can donate to type A to type AB. Type B can donate to type B and to type AB. Type AB can donate to type AB. Type O can donate to all ABO types. Blood transfusion must be studied, planned, and supervised by doctors. What are universal donors, universal recipients concerning the ABO blood system? Universal donors of ABO blood type system are the individuals of the type O. Type O blood does not have antigen A, neither antigen B in its red blood cells, and can be donated to individuals of any ABO type. Universal recipients of the ABO blood type system are the individuals of the type AB. Type AB does not contain antibody anti-A, neither antibody anti-B, and people of this group can receive blood from any of the ABO types. What is the type of genetic inheritance that determines the ABO blood group system? What are the relations of dominance among the involved alleles? The inheritance of the ABO blood system is a multiple allele inheritance. There are three involved alleles: IA, IB, and I, that combine in pairs to form the genotypes. Concerning dominance, allele I is recessive in relation to allele IA and IB. Between IA and IB, however, lack of dominance is established with the heterozygous IA-IB, manifesting distinct phenotype. What are the genotypes and respective blood types of the ABO system? Since alleles are IA, IB, and I, the possible genotypes are IAIA, that is blood type A, IAIB, that's blood type AB, IBIB, that's blood type B, II is blood type O. Is it possible to perform investigation of natural paternity, maternity, or brotherhood and sisterhood using ABO blood typing? By using ABO blood typing, it is possible only to exclude paternity, maternity, brotherhood, sisterhood, but it is not possible to conclude positively about these relationships. For example, if an individual has type O blood, II genotype, he or she cannot have biological parents of the type AB, IA, IB genotype, since necessarily one of his or her alleles has come from the father and the other has come from the mother. Another example, a couple of individuals of type O, II, in their turn, can only generate direct offspring of the type O blood, since they do not have alleles that condition antigen A, neither antigen B. Is ABO blood compatibility enough for the safety of blood transfusion? Beside ABO blood compatibility, the compatibility concerning rhesus blood system must be checked. In addition, it is of fundamental importance for the safety of blood transfusion, performing tests to detect agents of main blood transmitted infectious diseases like HIV, AIDS, hepatitis B, C, syphilis, Chagas disease, and many others. Any transfusion must be studied, planned, and supervised by doctors. What is the rhesus factor? Rhesus factor is a protein of the red blood cell plasma membrane that behaves as antigen in blood transfusion, triggering humoral immune response, antibody-based. Most people present the protein in their red blood cell and are part of the rhesus positive group. People that do not have the protein classify as rhesus negative. How are antibodies against the rhesus factor formed? Anti-rhesus antibodies are made by the humoral immune response. When a rhesus negative individual makes contact with a rhesus factor, this is recognized as a foreign or antigen. The primary immune response begins a small amount of the anti-rhesus antibodies, and memory B lymphocytes are made. In future contact with the antigen, there will be already circulating antibodies and memory immune cells prepared to create intense and effective attack against the rhesus factor. The origin of the name rhesus factor is related to the first researchers that discovered this blood antigen was in rhesus monkeys, Macaca mulatta. What is blood typing? Blood typing is the determination by means of tests of the classification of blood sample concerning blood group system, especially ABO system and rhesus system. How is the blood typing concerning the ABO system and rhesus usually done? In the blood typing for ABO system and the rhesus system, a blood sample is collected from the person and 3 small volumes of the sample are separated and dispersed on glass laminae or slides. On the first laminacelum containing anti-A antibody is dripped. On the second laminacelum containing anti-B antibody is dripped. On the third laminacelum, anti-rhesus antibody is dripped. If no agglutination reaction takes place in all of the laminae, the blood type is type O rhesus negative universal donor. If agglutination occurs only in the first laminae, the blood type is A rhesus negative, and so on. There are further methods of blood typing. Blood typing must be performed by a qualified technician. What are the inheritance and dominance pattern of the rhesus blood system? The inheritance pattern of the rhesus blood system is autosomal dominant. That is, heterozygous manifests as rhesus positive. The dominance is complete. R is dominant over small r. The possible genotypes are capital R, capital R, capital R, small r. Both are rhesus positive. And small r, this is rhesus negative. Curiosity, the rhesus factor is codified by a gene containing 2790 DNA nucleotides situated in the human chromosome 1. What is the logic of transfusion compatibility concerning rhesus blood group system? A rhesus positive donor can only donate blood to a rhesus positive recipient. A person that lacks rhesus factor, that is rhesus negative, can donate to individuals of rhesus positive and rhesus negative groups. What is the rhesus typing of the mother and of the fetus in the hemolytic disease of the newborn? In the hemolytic disease of the newborn, the mother is rhesus negative and the fetus rhesus positive. In this disease, antibodies produced by the mother attack the fetal red blood cells. The hemolytic disease of the newborn is also known as erythroblastosis fetalis. How does the immune process that causes the hemolytic disease of the newborn take place? In the hemolytic disease of the newborn, mother has rhesus negative blood. This mother, when generating her first rhesus positive child, makes contact, possibly during delivery, with the rhesus positive red blood cell of the child. Her immune system triggers the primary immune response against the rhesus positive factor. In the next gestation, in which the fetus is rhesus positive, mother will already have much more anti-rhesus antibodies in her circulation. These antibodies cross the placental barrier and gain the fetal circulation, causing fetal hemolysis, that is destruction of the red blood cell of the fetus. How can the hemolytic disease of the newborn baby be prevented? Erythroblastosis fetalis can be prevented if, in the first delivery of a rhesus positive child, a rhesus negative mother serum containing anti-rhesus antibodies is given to the mother in the first 72 hours after the delivery. Therefore, the administered anti-rhesus antibodies destroy the fetal red blood cell that entered the mother's circulation before triggering her primary immune response. What is the MN blood system? What is the pattern of genetic inheritance in the MN blood system? The MN blood system is a third system of blood antigens, also related to proteins of the red blood cell plasma membrane. The inheritance pattern of MN blood system is autosomal with codominance. The type of lack of dominance in which the heterozygous manifests a phenotype totally distinct from the homozygous. The possible phenotypical forms are three blood types: type M blood, type N blood, type MN blood. Thank you for participating in Blood Types. Please do like, share, and subscribe.